get started. I was about to say good morning, but good afternoon. For you, those of you that don't know me, I'm Sarah Velasquez. I'm the Communications Core Director for the K-INBRE. Hopefully we saw most of you last month when Dr. Wright gave his presentation about the INBRE. For a reminder, we do have social media. So we have Facebook, Instagram, Twitter. So feel free to join us there. We also have a website that has a plethora of information that might be useful to you, and about our program and funding opportunities. And we post the seminars on there and symposium information. So you might check that out also. Today, there goes Tim's chest again. Everybody's laughing. Okay, so today we have Dr. Fleming from Kansas State University. She's the coordinator there. She is going to present on how to keep a lab notebook. She has some great tips, and we are recording this. So if you have any questions, you can go back and watch it once I get it posted, or she will give you the opportunity to ask questions during or after the presentation. So we will get started now.
Thank you, Sarah. I'm going to try and share my screen here, and then I need to get it to full screen. Okay, so I'm going to talk about lab notebooks. Hopefully you've all been at least starting to keep a lab notebook. And if you're like most people, some days it's better than others. Most of my examples are coming from my experience. I'm showing you some of the good ones. I've got a lot of bad ones, so don't be afraid of asking questions about how you should do it better, because you can always do it better. But maintaining a lab notebook is really important because it's really the proof of how and what you did, what the data was, and how you interpret that data. So the purpose is just to maintain the records for your use as well as for other people's use. So you're keeping it, and your purpose is probably to be able to remember what you did on Friday by the time you've had a long 4th of July weekend, and where do you start on Tuesday again. But other people also are going to use that lab notebook. Your PI may want it down the road, a year or 2 or 3 or 4 later, saying, how'd they do this? Why can't we repeat it? Or I don't remember how they did it. Let's go to the lab notebook. In addition, it's a legal document to prove that you have the data you have for patents, and that you have the data to support that NIH grant. So NIH provides you, or provides the university, and therefore your PI, the grant money, but your notebook really belongs to that university, not even to the PIs, even though the PI actually gets charged with taking care of it usually. So it's really a legal document so that you can say, yes, I did it this way, and here's what, I did and how I did it. So in this notebook, you need to plan experiments. You need to have your data, but then to build on those results, learning from the mistakes made in the first experiment, how can I fix it and make it better in the next experiment? There's a lot of different types of notebooks. My lab is still very old school. We're using paper, but some of the labs have gone to electronic notebooks, and some are using a combination. I'll try to address that a little bit, but I don't personally have as much experience with the electronic format. Why does your PI care about it? Does everybody know what I'm talking about when I say PI? Your faculty member, okay? It's their insurance policy against scientific fraud. They can prove that something was actually done, and here's the data that was recovered from that experiment. So it's also that legal document for grant expenditures. And NIH can actually come and audit all of our notebooks. So it needs to be legible and understandable and not just hit and miss. Well, I did this and then I did this, and where's your calculations? How do you know that all of that is right? NIH could actually send someone to audit the lab and audit all the notebooks. I don't personally know of anyone having had that done, but that doesn't mean that they don't do it, especially when they're looking into various different legal problems that a lab could actually have. If you apply for a patent, or your faculty member does down the road, they've got the data and evidence of the invention there. So make it legible. We want to be able to use your notebooks down the road. Okay. Besides, if you go to grad school, you want to remember what you did 2 or 3 years ago, because you're going to have to write it up in your thesis. Okay. I'm going to address the electronic notebooks now, just because I don't know as much about them. And most of the basic information is the same for either of them. First off, the faculty member in charge decides what type of lab notebook the lab is going to use. So you can't just go deciding you're going to use an electronic one unless you've talked to your faculty member about it, your mentor. An advantage is that the electronic notebooks date and most times timestamp all the entries, so that you don't have to keep writing in, I did this on this day, dating everything. Some of them can actually provide initials of the person that did it. You can type things in and then insert data down below. It may have a phone app, but you may need paper copies for those real-time notes, especially if you're in a BSL 2 or 3 situation. You need to have paper, take pictures, and then go back and electronically insert it. But the information I'm going to provide down below or in the rest of this talk is going to apply to both, because it's going to be, what do you put in it? What needs to be there? Everybody knows they can write things in there, but what do you need to write? So as I've said, it's got to be legible. It needs to provide enough material so that someone else could come in and repeat that experiment. That means you need to define your abbreviations. I use CFG in place of centrifuge all the time, all throughout my notebook. But you need a list of those abbreviations that you use. You don't need to make microliters and micrograms, but weird things like my CFG, I need a list of abbreviations in your notebook so that somebody knows what on earth you're talking about. Most faculty members in the US and in Kansas are going to request that it be in English. They want a date, and you need to keep it where it's easily accessible. I go in and occasionally I'm trying to figure out who's going to do what next week, and I'm looking at their data going, where's the notebook that goes with it? What did they actually do here on a weekend? So I need to be able to find the notebook. And notebooks, generally speaking, unless you've gotten very special permission, do not leave the lab. They stay in the lab. They belong to the university. And every page should be used or crossed out and initialed. This is extremely important when you get into the good manufacturing process. But even in my lab, I don't want you wasting a page here or there because notebooks are expensive. So use every bit that you can. You're going to want enough information there to provide enough that you could write your poster methods from your notebook. Okay. You need to organize it. This is an office, but many times notebooks look like this with pieces of paper stuck in it and all over the place. No, 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 no. Despite the fact that I do it myself, I'll tape in little Post-its that I've scribbled on, but we shouldn't be. We should have it very organized so that you know what you've got. The first thing I recommend is having a table of contents. That table of contents, and I'll save a couple of pages in the front of my notebook. I'll put in the date and the title. And if I'm doing it multiple times, which most experiments you are, the first time I'll say nitric oxide assay. And then the next time I'll say nitric oxide assay again on J7s or whatever. But the first one, I'd go back and say, didn't work. And I'll put a little comments, or I will highlight things that actually are really good and beautiful experiments that I want to use for publication. Okay. You also should keep a list of where your protocols are, because you got to know which protocol you use. Did you use, we have trisol protocols for making RNA, collecting RNA from cell lines, and a little bit different from tissue. Which protocol did you use? Identify where it is and have those in there. So for every experiment, this is what they recommend that you have. You won't see all of this in mine, I can guarantee it. But it's what we aim for. We need a date and a title. That's fairly easy. Usually they recommend that you give 1 or 2 sentences of a justification of the experiment, your logic, your rationale, a reference, something like that, and your hypothesis. Then your methods come next. This is pretty straightforward as to the order, and you want to write it as you go, because sometimes you plan to incubate something for an hour and it goes an hour and a half. So you need to make sure that that actually gets written down. Or you plan to incubate today at room temperature, but they've turned off all our steam, so all our rooms are about 60 to 65 degrees today. Well, that's something you should write down, okay? Then you want to have a summary or a conclusion of what your data showed and the samples that were collected. Where are they kept? How are they being kept? That type of thing. We'll go into some more detail in a minute. And who helped with the experiment? On your posters or in your papers, you're going to have an acknowledgement section. If you've written the experiment and written down, Jen took my pictures, I verified my calculations with Nathan or whoever, that way you know who to acknowledge in your acknowledgement section. Okay, protocols. You usually start with a protocol, at least in my lab, people are given a basic protocol, but I like for them to write it out the first time. And that way they can make notes along with it. They can indicate additional information. If you're going to do that nitric oxide assay or whatever, a second and third or fourth, a 10th time, you can say in that one, oh, I used the nitric oxide protocol on this page of my notebook. That's another thing. I think I skipped that earlier. Pages should be numbered so you know how to find things. Every time you write down your beginning and ending times of your long incubations, that way you aren't saying, well, it was about an hour and a half, or it was about an hour, but it was really an hour and 15 minutes. If you have multiple machines of the same type in your lab, you should identify which machine. Not that you need that for writing a paper, but if something breaks and something's getting weird data, you can figure out which machine it was on at first, or easily from the notebooks. Record all your calculations. Show all your math. Don't transfer it. That's not legal. I know many of us write it on a scratch paper and then transfer it over, but carry your notebook over to the cell counter and write down how many cells you counted. Or if you're going to write it on scratch paper, tape that scratch paper into your notebook so you're not rewriting. We make mistakes along the way, write them down. We all do. Yep, I planned to do this. I used the wrong antibody. I used the wrong water or whatever. Identify the problems in your procedures. Any lapses of data collection. I overslept. I didn't get here at 24 hours or at 8 hours or whatever time, 12 hours from when you started or when you finished it, you were supposed to collect it. I didn't get there in time. So write it down and explain it. So maybe the results come up different. You've got an explanation. If you're using this protocol that you've got set, make notes on it if you make any changes. You dropped the tube on the floor and you had to do something, so you estimated how much was left in the tube. The reagent was only 50%, or I've done this one, made 75% ethanol and wasn't thinking and made it 75% water. Doesn't work, so don't make that mistake, okay? Put down these problems that you run into. For the methods, when you're actually writing them, first time, lots and lots of detail. Then as you make repeats of the experiment, you can make changes, or you don't need to write as much if you're following it exactly. Show all your calculations. Don't say, I counted 152 cells and I ended up with 5 times 10 to the 4th cells per mil. 152 cells and 5 times 10 to the 4th. You need some logic in there so that everybody else knows what you did. That means you need to include the units. Label all your figures and your calculations and make it legible. I get in a hurry and I'm scribbling many times so that I can't even read it the second time round. So be careful and make it legible. Specific details. If you have reagents, where'd you get it? What's the product number? What's the lot number or the expiration date? And where'd you find it? Was it in the refrigerator when it should have been in a -80? How was it stored? Solutions. How are they made? Did you dilute it from a 10x stock or did you make it from scratch? And where's the recipe? What type of water did you use when you diluted it? Did you use tap water? Did you use DI water? Did you use the really super duper good water? Put in a recipe, put it in the protocol. A lot of people are using cells. Okay, so you'll say, I use J7s, but the full name is J774.8.1. Okay, so write out that full name because there are actually J774A.1. There are J774Gs. There are lots of different types of J7s, and my lab at least has multiple of them. Where'd you get them? Were they frozen and you thawed them, or were they already growing? What passage number are they? Did you grow them in the traditional media, or did you change it? If you're doing washes, how many washes? How'd you do it? How much volume? Did you use a squirt bottle and fill a 96 well plate, or did you use a actual plate washer that puts in a set amount of volume? What type of instrument did you use? For centrifuging, what speed on what centrifuge? As I hope many of you know, most of you know, the speed, how many RPMs, is not the same as how many Gs from one centrifuge to another. If it's got a rotor this big or this big, it's going to change. How long did you centrifuge for? How long did you heat? What was the heating rate? Did you vortex it along the way? How long did you go for various things? If you're running gels, what gel percentage did you use? What type of gel was it? Any of those details that you can come up with, add, okay? That'll make it much easier down the road when you're trying to make your poster. Here's an example of one of mine. I was looking for contamination in these antibodies, and I checked the isotypes. I made these dilutions. I wrote this out ahead of time so I could keep track of what I was doing. I knew I was going to make a 1 to 10,000 dilution in carbonate buffer, but I didn't know what the pH was going to exactly end up being. So I left that blank and I put that in when I made it. And I diluted these guys. This one came from Mike and it was at this concentration. I needed to make a 1 to 100. So, and then I checked them off. I got all that ready and I incubated at 4 degrees overnight. The next day I added this, then I washed, and these I did at various different times the following day. So put in as much information as you can. Because I had written this out ahead of time, I went through and checked it off when I had actually completed it, so that I knew where I was and I was keeping track of how I was doing things. Then you get data files. Your notebook should know where the data files are stored. Are they on the general lab computer? Are they in a folder in inside your folder? Or where do you find them? We don't store things on USB drives. They get lost. What's the name of the file? Add that information to your notebook. We talked about adding and recording names of the people, so we've got it for acknowledgements. If you're using animals or humans, you've got even additional information. If you're using animals, keep the cage card with the experiment, or at least write down all the information. Have all that information there. Second time around, you can refer to that first protocol. You want to record the start and stop time of your long incubations and record any changes that you make. If you need to rewrite the whole thing, do so, but then call it a new protocol and put it in that table of contents. So basically I'm saying record all the facts. All the data needs to go into the notebook. All your raw data goes in, and it doesn't matter if it's a good experiment or a bad experiment. You're going to need to record it while you're doing it. If you're going to, wherever that is, check things off and have times written down, that's going to mean recording it as you go. Haul that notebook around with you. Don't leave it at a desk and go do stuff otherwise. Don't remove pages. Record your facts, not your opinions. I fail at this one as well. If nothing happened in an experiment, you can say nothing happened, but you're not supposed to say it will never work. You might say that verbally, but you're not supposed to write it down. Sometimes I put a big X on things and get irritated, but be honest, put it all down. Okay, are there questions before? Well, no, do you have questions now before I go on to making mistakes and changes?
I think there's a good comment that was made in the chat about the write as you go being very important, especially for publications.
Yes.
So that, and I can verify that with my dissertation research, that going back and trying to write that later, it was really important, the notes that I took. So
I think every single person that's worked in the lab for more than a month can verify that you don't remember what you did a month ago because you've been doing other similar things. Write it down, and write it down as you go. Not that night, not at the end of the week, because you won't remember exactly what you did.
And this, I will also add that this is super important for a reviewing process. So more and more now, people are asking questions about, like, you know, detail of procedures, something that, well, we obviously want to show everything and try to add everything, you know, public. But sometimes, you know, a reviewer or someone might just contact you and asking you more detail or some pieces of information or material that you may not have put in the original submission. So I think everything that you can have to kind of secure and you know, protect your work, and I think that's really important. So that was a great point.
I would like at times, and I suggest it to my students, they don't really appreciate this suggestion, but we can just tie it around your neck, and that way it's with you to write it down. So I won't make you do that, but it's that type of memory. Have it with you all the time. Okay, what if you have a protocol and you made changes to it? So I've crossed out this. I highlighted it up front saying, this is what I'm going to do. And I wanted to make sure I remembered it, but it's not what I actually did. This was not the right concentration, and things changed. So here I ended up crossing things out, writing notes, and changing things. For FC1, I didn't use the 20 nanograms per mil because I didn't have enough. So those two got crossed out. Cross it out and take it with you. Do's and don'ts, overall do's and don'ts, keep it legible and orderly. You can write ideas for your next experiments because they're usually coming from the current experiment. Have enough for someone else to come in and replicate it. Many times I have students trade notebooks at a lab meeting and say, choose this experiment. Here, here, here. We just arbitrarily open them and say, can you understand what they did? Could you go repeat it? Now they're at least familiar because they've been in a group working together. If they can't repeat it and there's not enough there, then you know you need to add more to it. Have your abbreviations defined and include all the details. Modify it by a line through it. Don't erase it. Don't totally try and scribble it out. Let it be obvious what you're changing, okay? Don't rewrite from scraps of paper. Don't skip pages and leave blanks. Don't remove originals, and no whiteout. Today's world, I don't think anyone's using whiteout anymore, but you still can't use whiteout, okay? Okay, current good manufacturing practices. These are practices that are regulated by the FDA. You frequently have them in business or industry. And if a lab is patenting things and moving it towards clinical trials or something like that, you'll need to start following these. They have a lot of changes that occur as FDA makes changes, as any government agency changes, it's going to have changes. And that's why it's called CGMP for current. It's used for final drug products, medical devices, vaccines, and everything like that, so that we have everything uniform. You want a paper trail, data trail for everything. That includes test tubes. So the test tube labels or a picture of those tubes can be inserted. Everything gets followed. These can be in notebook form, or sometimes they're actually in a worksheet form, and you just fill out the worksheet for every experiment. They usually have a signature on them, at least one, sometimes more than one, and that makes it an official document. Examples are up here at the top. They didn't do that, so they initialed and dated. You can use a dash if you didn't do something. And if you're adding in that little post-it note that you did your calculations on or took your numbers on, draw these lines across it so that it can't be removed without it being obvious that something's missing. Okay. Other things that are general CGMP things, fill it out in real time, a single line to cross something out, no whiteout, no sticky notes, no anything else, no write-overs. So here they crossed out the 2, but it kind of looks like it could be an 8. And here it looks like they made a 7 into an 8. You can't do that. Fully explain your calculations. So it's very similar to what we do. It's just more controlled and regulated. And every business industry is going to have its own specific way of doing it. So learn what they want from you in keeping a lab notebook. And with that, I'll stop and see if you have any questions or comments. Again, students, you've been keeping lab notebooks. What questions do you have? Okay, I've also put up here 2 YouTube videos. The first one's almost an hour long, so we obviously can't watch it. It's about misconduct that involves not only, it's a misconduct video, but it also involves notebooks. And then a second one that's like 20 to 30 minutes long. And they're both different. I thought I'd share my slides with you, and you can use them and watch them later if you'd like.
Sherry, could you possibly copy and paste those into the chat for us?
Probably. If I get out of,
or whenever.
I can stop share, and I have to get out of my screen there. Yes. There we go. There you go. I've used those for faculty members. I've used those for some of the RCR training type things. So.
Hi, Dr. Fleming. I have a question.
Yeah.
What do you do if you're working on multiple things at a time?
So there's two ways you can address that. And a lot of that depends on your PI. I'll give you my opinion and other PIs can chime in. I will write part 1 up here, not save space, because I might need that space. I might need more or less than that space, but start the next one. And then on the next page or the next week, I'll refer back to that page and that experiment number in our lab notebooks. That is an advantage of the electronic notebooks, where you can actually go in and insert day 2 and it's got its own timestamp. But in paper, you could try and leave a space. My problem is you don't know how much space to leave, because if you just have to do like the next couple of days and you do something like I did, I put it in my protocol and I checked it off and put the date. If you can write it up ahead of time, that works. But if you coded plates for an ELISA and you're going to get to it and you don't know what the samples are yet and haven't got all that done, you're going to come back to it maybe next week. I'd just say, I coded them this day with this much like this, and I'd reference back to that in my next log of it. It makes it a little bit messier. You have to go back and forth, but for me, it works better. Do other PIs have an opinion on that? Or anyone else? Tim, you're sitting there looking at us. How do you do it?
Yeah, I like the comment that just came up about maintaining chronology. I mean, that's the kind of thing that I try to stress, is to just think about it as a chronological record. And you may have to write, transition statements, and I'm transition, like I just put this in the incubator for 4 hours, and while that's going, I'm going to go work on this other piece. And just writing a little transition statement in there can kind of help you make sense of it. But yeah, the idea of referencing back and keeping in chronology, I think that's an important aspect.
There are some times, as I showed you in that one, where I've written it out ahead of time and then come back and checked it, and but that's a protocol that we used routinely, and so I just stuck the whole protocol in there. In my notebook later on, I would say also did those steps in, but I stuck that protocol in. So. Other comments? Questions? It seems easy right now. But is it that easy to put everything in so that your neighbor can figure out what you actually did? And then if they can figure it out, how about if I came and looked at it? Could I repeat it? Because I don't know what you're actually doing. Okay.
Sometimes sometimes putting a few more seconds to write something can just save you just hours of interpretation, like later, again, like when you're writing manuscript or preparing your poster, and that can just save you so much time later. So better just put, and I talk about it because I keep making that mistake sometimes. I'm thinking, well, I will remember that tomorrow, and it's just gone because there are so many things going on.
I think we all have that problem, and it gets worse as you get older. Anyone else? Have any of the students had to troubleshoot a problem that isn't working right and tried to use your notebook and it failed? Or am I the only one that has failed notebooks? Sarah, our students are absolutely amazing.
That's good to know.
That's all I have. So,
Well, if there aren't any more questions, then Dr. Fleming would like to thank you for presenting to us today. I will get this video up next week on our website in case anybody wants to go back and watch it. And if anybody has any questions, you're welcome to contact me or Dr. Fleming, and I'll put you in touch with her. Otherwise, then we'll see you next month for our next seminar. Thank you.
thanks.